Good morning, everybody. Today we will understand genetic code. Code is basically the term which refers to the set of three base code words or the codons in the mRNA that stand for 20 amino acids in the proteins. That is, in simple terms, it is three bases which will code for one amino acid. Three bases coding for one amino acid. In the same way, we have various codons which will code for 20 amino acids, and these codons are present in the mRNA. So the set of these codons is what we refer to as the genetic code. Here we can see the table which represents the genetic code. For example, U, three different bases, U. It codes for phenylalanine. In the same way, the another code UUA codes for leucine. At the same time, another code UCU codes for another amino acid serine. So a set of three bases of the mRNA which code for a particular amino acid is called as a codon, and a set of all these codons put together is what we call it as the genetic code. Now, before going into the salient features of the genetic code, how can you write this genetic code easily? So let's write four bases, that is U, C, A, and G on your left hand side, and U, C, A, G in the rows, in column and in rows. Now, what we can do is U of the row and U of the column, and the UCAG on the extreme right side. U, and an U, and a C. U, and an A. U, and a G. So here is how we have filled the table. Can you make a note of it? Here is U on this, and U this side becomes a codon. Next codon U, and C. Next codon U, and A. Next U, and G. In the same way, we can fill the next box. Here UCU. So the first two letters will be the same. First two bases will be same. UC. So you can fill UC for UC, and then UCH. That becomes your genetic code. Here, if we move to the next box, the next is U and A. So the first two bases will be and the last base will be UCAG. So this is how we can fill the whole of the genetic code which codes for the 20 different amino acids. And as you can see here, not just a single codon, there could be more than one codon which can code for one single amino acid, right? Apart from that, we have other set of codons which we call it as stop codons like UAA, UAG, and UGA, which basically are the signals to stop the process of protein translation. So once these codons are seen on the mRNA, the process of translation is terminated, and hence these codons are called as stop codons. So let's look into the salient features of the genetic code. That is, its near universality among all forms of life is compelling evidence that life on Earth arose from a common ancestor and makes it possible, for example, to express human genes in E. coli. That is, in the beginning, the genetic code was considered that it is almost universal in all the organisms, and hence was considered that all the organisms have a common ancestor. But that's not the case true right now. There are different genetic codes seen in different organisms. But when the genetic code was discovered, it was seen that most of the organisms which were studied had a similar genetic code. So, when they had similar genetic code, people tried studying what are the four bases that can occupy each of the three positions in the codon, and can they could derive at how many codons are present. So, what was it done? So we know that there are four possible bases, that is U, C, A, G. Why U? Because we are speaking about mRNA. So U, C, A, G. There are four bases that can occupy each of the three positions of the codon. That is, U can be present in the first position, second position, or third position. So all of the four bases have equal opportunity to be present at any of the position in the codon, first, second, or the third position. And hence, there are 4 to the power of 3, that is 64 possible codons. This is how they derived at how there are 64 codons possible, just because we have four bases and each base can be present at any position, the first or the second or the third position of the codon. 4 to the power of 3, there were possibly 64 codons. Of these codons, that is of the 64 codons, 61 specify amino acids. That is, 61 of them code for 20 amino acids, which I showed you in the table. And the remaining three, there are three codons which are called as stop codons, that is UAA, UAG, and UGA are the stop codon that instruct the ribosome to cease polypeptide synthesis, that is, stop translation and release the resulting transcript. So there are totally 64 codons, out of which 61 codons code for 20 amino acids, and three of them are called as stop codons. These codons are otherwise mentioned as nonsense codons, UAA, UAG, and UGA. These are the stop codons. All but two amino acids, methionine and tryptophan, are specified by more than one codon, and three, leucine, serine, arginine, are specified by six codons. So, if we look into the table, the amino acids methionine and tryptophan are actually coded by more than two uh, codons. We can go back to the table. If you can see here, methionine, AUA, and AUG. Here you can see methionine. You can see tryptophan here, UCG. So these are the ones which are coded by the two codons, and you can see the other amino acids like leucine. You can see that leucine UUA, UUG, CUU. So there are six codons which are coding for the same amino acid leucine. So there are few amino acids which are coded by more than two codons, like your leucine and serine and arginine. There are more than two codons. The codons differ in sequence only in their third nucleotide. So if you look at the codon, it has three bases. I've already shown you in the table that the first two bases will be same because you of this, you of this. Only the last base is differing in each of the box here. So that is what is the next salient feature, that is the codons differ in sequence only in the third nucleotide. And then most codons specifying non-polar amino acid residues have a G in the first position or U in the second position. So if we again look at these codons, non-polar amino acids. The codons which code for non-polar amino acids will either start with a G or a U. And many amino acids could be specified by more than one codon. Such a code in a term borrowed from mathematics is said to be degenerate. So, since more than one codon is coding for one amino acid, there is a mathematical term which is used when there is one codon coding for more than one amino acid, which we call it as degenerate. Hence, it is also called as genetic code is degenerate. We have a genetic code. It is non-overlapping, it is comma-free, and it is degenerate triplet code. Triplet because there are three bases. We'll understand why we call it as non-overlapping, and we'll also understand why it is called as comma-free. So these are the salient features of genetic code. This can be asked as a 2-mark question or a 4-mark question. What are the different salient features of the genetic code? So we are supposed to write that how there was 64 possible codons and 61 specify amino acid and 3 are stop or nonsense codons, and all but two amino acids, methionine and tryptophan, are specified by more than one codon, and three, that is leucine, serine, and arginine, are specified by six codons. The codons differ in sequence only at the third position. Most codons specify non-polar amino acid residues have G in their first position or U in their second position, and the amino acids it's can be specified by more than one codon. Therefore, it is called as the genetic code is degenerate. It is non-overlapping, it's comma-free, and since it contains three bases, it is called as a triplet code. So now we'll see why is it called as non-overlapping or what is non-overlapping. It's a non-overlapping code. Each base is a part at most one is a part of at most one codon. So let's see this is the mRNA. So here we have in the mRNA AUG. So when we say it is non-overlapping, the codons which are formed from this example mRNA is two codons. One is AUG, the other codon is UUC. So that is what is called as non-overlapping. If it is an overlapping code, then it should be AUG, UGU, GUU, UUC. So many codons should have formed. But the triplet codon is a non-overlapping codon. Hence, if you have six bases, then it will form only two codons. If you have nine bases, it will form only three codons. So it is non-overlapping. One base may be part of two or even three. In a non-overlapping code, one base may be part of two or even three, but in a non-overlapping, it is only one in one position. Okay, so what we are saying is genetic code is non-overlapping. So it is AUG and one first codon is AUG and the second codon is UUC. This is about non-overlapping codons. So there was an experimental proof to show that it's a non-overlapping codon. So Frankel Conrad laid it to rest with the following line of reasoning that if the code is non-overlapping, a change of one base in an mRNA, a missense mutation, would change no more than one amino acid in the resulting protein. So, what was his? These are theoretical proposals. So, what he proposed is, let's assume that if the code is non-overlapping, that is, it has only two codons in the given example AUG and CUA, it should form AUG CUA. If it is non-overlapping, if I change one single base, what would happen in let's imagine we are changing one of it. a change in the fourth base. Let's say the C would change only, uh, let's say there is a change in the fourth base. So see, actually, if it is non-overlapping, it should form AUG CUA 2. If there is a change, let's say C is changed to A, then it will form AUG AUA. Only one change and one amino acid. But if it is a non-overlapping one, then if it is a overlapping one, then what would have been there? AUG, UGC, GCU, CUA. So if there is even one base part of three adjacent codon. So you have that C which is present in UGC, GCU and CUA. So if C were changed, up to three adjacent amino acids could be changed. So one change, if it is overlapping, one change in a six bases reading frame of an mRNA, there could be three different amino acids coded. But if it is non-overlapping, where from six there are only two amino acids which are formed, six bases, two codons, only two amino acids, then if there is a change, there will be only one amino acid which will be changed. Okay, this is what was the proposal. But when the investigations introduced one base alterations into mRNA, later they introduced, this was hypothesized, and then what they did, they introduced one base alteration into mRNA from tobacco mosaic virus, and they found these never cause changes in more than one amino acid. So when they had a set of six bases, one base is changed, then they saw only one amino acid was changed. Therefore, they came to conclusion that the genetic code is non-overlapping, because in the example we saw that if it is non-overlapping, one amino acid will change. If it is overlapping, three amino acids will change. So, since there was only change in one amino acid, it was concluded that the genetic code is non-overlapping. Next one is no gaps in the genetic code. So, what is this no gap? So, if you look at the reading frame here, AUG, CAG, ACG, if translation starts at the beginning, the codons will be AUG, CAG, CCA, ACG. So, these are the four codons. If we insert an extra base, so let's assume we are at the third place, we are inserting an extra base right after base U, then what will we get? AUX, GCA, GCC, AAC. So there is another which is left out, which will not code because there is no other two bases. This would translate into this, okay, correct? So the extra base changes not only the codon in which it appears, but every codon from the point on, because see, we have changed one, AUX, then GCA, GCC, AAC. Before it was AUG. AUG, instead it has become AXA. Then we had CAG. But what happened here? GCA. Because it has moved, there is a shift in the frame. One, we have introduced one extra base, correct? So this would translate into totally new form of the protein. The extra base changes not only the codon in which it appears, but every codon from that starting point. The reading frame has shifted one base to the left. So what happened? The reading frame has shifted. Whereas C was originally the first base of the second codon, and G is now in that first position. So you can see that it has changed. What has changed? The G is now in that. C was originally in the first base of the second codon. C was the first base in the second codon. But the same C has changed to the second position in the second codon after we have introduced an extra base. Okay. So if If a code is with commas, it would be one which codon is flanked by one or more than untranslated bases represented by Zs in the following message. So let's imagine it has commas. Then what it would be AUZ, comma, CAZ, comma, CCA, comma, ACG, comma. So Zs here are representing comma. Let's imagine. So deletion or insertion of a base anywhere in the message would change only a single codon. Let's say we are deleting something. The comma Z at the end of the damaged codon would be then put the ribosome back on the right track. Thus, addition of an extra base to the first codon would give the message AUXG, comma, CAG, comma, CCA, comma, ACG. So the first codon AUXG is now wrong, but all other codons till neatly set off by Zs would be translated normally. So what happens if it was having something like commas, which would tell that here is the finishing of one codon, it would have remained the same like the first frame, no shifting of the frame. So there was an experiment which was conducted again. Francis Crick and his colleagues treated bacteria with acridine dyes. So what was the experiment? They treated the bacteria with acridine dye that usually causes single base insertions or deletions. It basically causes insertions or the deletions, that is single base. They found that such mutations were very severe. The mutant genes gave no functional product. So what happened is, if it was having commas, even on insertion of one or deletion of one, what should have happened? Just one amino acid should have been coded differently. But what they saw was it was totally a different product and it had no function. So, what would you expect from the experiment? That the genetic code is a commaless code with no gaps. Hence, there was a new product which was formed even with a single insertion or a deletion of the base. Base insertions or deletions cause a shift in the reading frame of the message that persists until the end of the message. So, what will happen? One base change will also cause a lot of change in the frame, and hence the function of the product was changed. And Crick found that adding a base could cancel the effect of deleting a base and vice versa. He also did another experiment. So, first deletion and then addition. So, they were doing different types of experiments of deletions and insertions. And what he found that adding a base could cancel the effect of deleting a base. So, let's say we have added one base and then we have deleted. The effect will be removed. In the same way, you delete first and then add, then the effect will be removed. Here you can see wild type CAT, 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 CAT. We added a base, it became